All right. So let's talk now about the BIGR eye. So before to talk about the BIGR eye, let me just very quickly introduce the team. So the BIGR eye results from a joint effort of Theo van Wolsten and Stefan Clay. And working on full time, there is Luisa, there is myself and Konstantinos. And besides that, we had a number of students this year doing internships, bachelor thesis, master thesis from different institutions in the Netherlands and also from abroad. And that's our international network. All right, so by now you have already realized that we are going to talk about human eye, right? And but before to talk about that, and just to break the ice a little bit here, and today is a cold day, let's just say some cool facts about the human eye. So in fact, you see with your brain, not with your eyes. So your eyes basically just function like a camera, capturing light and sending then that data back to the brain. And if the human eye was a digital camera, it will have 576 megapixels. And the fact that we have two eyeballs give us depth perception. So comparing two images allow us to determine how far an object is from us. So the only organ more complex than the eye itself is the brain. But about half of the brain is dedicated to vision and seeing. And this is because 80% of all learning comes through the eyes. And this is only possible because we have 7 million cones, which help to see color and details. And we have 100 million of cells called rods, which help to see better movements and to see better in the dark. And so far, I hope that I have already convinced you that the human eye is the most interesting organ in the human body. But I hope that you don't have omatophobia, because that's the fear of the eyes. So this is how an eye looks like. So you have the anterior part and you have the posterior part. So the anterior part is responsible for the diffraction of the light. And then the posterior part basically does all the magic. So basically gets the light information and converts that to a signal that goes to the brain. But at this posterior part, which is called the retina, a lot of diseases can happen, which will impair your vision or eventually lead to blindness. One of those diseases which affects our society is called glaucoma, so the silent thief of sight. And this is a very important disease because you don't have early symptoms of the disease. You just start to have this kind of tunnel vision, but you don't realize that because your brain will just reconstruct that information. And when you realize that, it's already too late because you cannot restore that vision, and you can even get blind because of that. So that's why it's very important to do retinal imaging. So, but as in any imaging modality in the human body, there is always the good, the bad, and the ugly, right? But here the ugly will be the solution. But when it comes to the good, so the good thing is that it allows early diagnosis, retinal imaging. So it's much more affordable than other imaging modalities that we have here today, like MRI or CT. And it's non-invasive. So in the majority of the cases, there is no need for any contrast agents. And the bad thing is the diagnosis nowadays is mostly done by visual inspection. So there is a big lack of tools to help the clinicians. And in addition to that, so there are also still a lot of differences between the current commercial devices and also evaluation scales and acquisition protocols. So given that, it's very difficult to observe small changes by visual inspection. And a lot of times these images do not have enough resolution. So the solution here is to develop new image processing and artificial intelligence-based tools to help for a better diagnosis and prognosis in clinical practice. And that's what we do here at BIGR Eye. So we have a number of projects that I would like just to succinctly talk about, but then for more details, you can always join our channel. So one of the projects is about glaucoma classification using deep learning techniques based on multimodal retinal imaging. And here we have we'll have as input imaging modalities, so different imaging modalities which give us structural information, and we will also have as input functional imaging modalities which are related with the vascular information at the retina. And then we will combine that in a convolutional neural network and then do the prediction and the classification of different levels of severity of glaucoma. So this is a project in collaboration with Kyle Leuven from Belgium and the University of Porto in Portugal. Also in line with this, there is this project which basically will make use of one of the biggest glaucoma data sets in the world, which is the Leuven eye study. So it's 600 subjects acquired with data acquired over 15 years now, and there are a lot of measures from that data set, since OCT, OCTA, ultrasound, etc., etc. So in this project, we will apply supervised machine learning methods to do the classification of the disease and also the regression analysis, but we will also look at unsupervised learning, because a lot of times even the classification within the different types of glaucoma is not clear for the clinicians, and we want to compare, given all of these measures, what's the difference between this unsupervised learning and what the clinicians are doing at the moment. Once again, so this is a project in collaboration with Kyle Leuven and Porto. Another interesting research line that we have is related with adaptive optics. And this is very exciting because adaptive optics is a technique that was brought from astrophysics to ophthalmology. And basically, it helps to, it corrects for the aberrations of the light between the object and the image that we have. And so what we are doing here is also applying this same technique to the human eye to correct for the aberrations that exist in the cornea and the crystalline lens in order to get a very sharp image of the back of the eye. And with that, we can see very clearly all of those photoreceptors that I was just talked at the very beginning. And in this project, what we will be doing is an automated way to do the segmentation of all of these photoreceptors with deep learning and then provide this information to the clinician. So to extract biomarkers from that and provide to the clinicians. And so this is a project with Cansva, which is a hospital in Paris, Imaging Eyes, a company also in Paris, and of course BIGR. And also with these kind of images, we have another project which, instead of the photoreceptors here, we are focused on the vessels, and we want to extract information like the inner diameter and outer diameter of the vessels, because all of these biomarkers that we extract from these images then can be related not just to pathologies that exist in the eye, but cardiovascular pathologies. So the eye can also be used kind of window to the cardiovascular system. Another very important project that we have is focus on the segmentation of the retinal layers. And this is very important because changes on the thickness of these layers will be certainly related with different kind of pathologies. So in order to provide the thickness to the clinicians, we need to have a very accurate segmentation. So that's what we are doing in this project. And this is a project in collaboration with Imaging Eyes from France, a hospital in Denmark, Region H, and the clinic here at Erasmus MC. And we also have a project focused on the anterior part of the eye, and this project basically will focus on the cornea and will extract features from the corneal tissue. So what you see here, it's just basically an air path of the cornea, and the cornea flattening in and flattening out. And based on that flat in and flat out, we can extract features to classify the cornea as normal and keratoconic eye. And besides of that, we'll also look at the values of the intraocular pressure, how that relates with these properties that we get from the image. And this is a project within collaboration of different groups within BIGR and also with Polytechnic of Wrocław in Poland. And the last project is focused on the segmentation of the vascular tree on fundus images. And in this project, so what we aim here is to, once again, with deep learning, so segmentation of this vascular tree, and then to extract features from the vessels, and then link those features from the vessels to dementia. So this is also a very exciting project where we will basically use the eye as a window to the brain. And this is a project so in collaboration within the clinic of Erasmus MC. So very succinctly, that was all I wanted to say, but yeah, I would like to tell you much more about the projects. And if you're interested, please join later on the Channel C eye imaging. Thanks.